can see the inflammatory response like this, hemorrhage. And also sometimes you can see fistula. So what is fistula? I also have here. It's the connect between scrotum. I have just add this one. Scrotum and also testis. So once have infection, sometimes the way to repair also can join some parts together. So you can see like the joint sometimes. And also here the common organisms be the positive agent of this disease usually is E. coli, Escherichia coli, and also Neisseria gonorrhea. That's why you can see the pus here in male and also female. And also chlamydia, Phacomyces, and also, this one is also nognatium. So usually sometimes like when people get infections, sometimes can get both infections. So we say it's quite common to get both infections. When you see pus, you also can like need to check whether which bacteria or which microbes. And also sometimes is the Trichomonas pallidum. This one is syphilis also. That is the causative of the acute otitis. And to see whether it's acute or the chronic otitis is also depends on the immune response that come to help. Actually, in acute otitis, you can see the so many like the several neutrophils reach in the area, and sometimes like the infection occur until the tissue damage. You also can see necrosis somewhere that you please take a look whether you can see the pyknosis, cardiolexis or cardiolysis in the area. That is necrotic area. And in the seminiferous tubule, if you take a look here, also can see the pink color. So because in any inflammations, also try to have fibrous tissue come to like repair in inflammations, and also the lymphocytes infiltrate here is also chronic inflammation. If you take a look, fibrous tissue like this and also lymphocytes instead of the neutrophil. So this one is chronic inflammation, chronic otitis. Checking where is you can find a neutrophil rich or lymphocyte rich. And I think our sample is this one. So focus in the lowest magnification first. So you can see in the testis, and along here is the lesion. So focus like closer and closer. You can see like here darker color. You can see maybe in the neutrophil. Oh, I'm so sorry, the nucleus. So the where that have the nucleus, maybe is inflammatory cells infiltrate in that area. That's why like look closer and closer. You will see even whether it's maybe neutrophil or the lymphocytes. Please check. And the next one is the example of the tissue necrosis. So this one is the gangrene. Gangrene, the cause of gangrene is lack of blood supply, lack of oxygen to the area, right? That's why we have the cell death. So here, if you take a look in the blood sample, outside you can see the black dark color. Maybe you suspect, maybe hemorrhage or even the cell death. You don't know until like have the cross section. That's why you need to check at the cross section. Here is selosa. Selosa is with the surface that's outside. In the lumen we call mucus mucosa. That's why expect that at the selosa side you may see hemorrhage. And here, hemorrhage here. And as I told you right here, that is the lumen. So it's mucosa side. And also outside here is selosa. You can see hemorrhage. So please take a look at the lowest magnifications. Again, you can see normal with us here, and then until you see somewhere else here, the structure is already be destroyed, the tissue damage. So I think you're familiar with this one. So please take a look again because we have worksheet to do today. And the last one, we don't have the gross specimen. We cannot take the gross specimen like this, right? Because this one is the bacteria. Even the structure of it look like hyphae, it look like fungi, but actually it's bacteria. This one again, facultative and aerobic bacteria. So it means they are anaerobic bacteria, but sometimes they also can stay in the area that with or without oxygen. That why can penetrate deeper and deeper to any area of our body, right? Especially at the oral cavity here, you can see until like this. And this one, you can see the lot shape like this, look like fungus, like large network. And under microscope, this one is chronic abscess. And thinking about abscess, thinking about neutrophil niche, but this one why is chronic? Because this one actinomyces species, so it grows very slow. So the characteristics look like fungi. So once they grow slower, so our immune response also try to eliminate, like grow, eliminate, grow and eliminate. So until it's like chronic, so taking longer time. And also here, if you take a look, it looks like fungi, but this one is colony of bacteria. Like so many colony of the lot-shaped bacteria is here inside like this. And also around here, you can see the lighter color. So this one is fibrin. Fibrin is because usually when we have infection, we would like to encircle, have the border wall off the bacteria. That's why the things that can wall off bacteria is fibrins of red blood tissue. So its fibrin is the protein here, protein rich, try to wall off bacteria, colony of bacteria. And after this fibrin area also have neutrophil niche. So that's why it's still neutrophil because it's abscess, but it's chronic. You can see until like this. And our specimen is really this one. So you can see so many like colony of bacteria. And please like take a look the fibrin, and also like you can see a lot of Mickey Mouse because it's neutrophil here. And the next one will be fungi. So Ajahn Ying will talk about the fungi. Because like to save time when you go to take a look at the lab, so we don't need to like, we would like to get everyone concentrate at now, and then later we'll take a look.
Thank you, So we have got specimen of the lungs. I think we bring one sample today, maybe this one. It's actually the same lung, but they cut it half. So then you see this as the two pieces. Okay, so you can observe the fungal granuloma as well in this case, similar to TB granuloma. The appearance might look similar. For example, here you can see like pale color of nodule, small nodule spreads into the lung. As mentioned earlier, that look at just the gross or chest X-ray cannot tell specifically what it is, whether it is TB, fungal infections, or something else. You have to look at it under the microscope to take sample and do staining and look at it under the microscope. What exactly is this infection? And as you can see, the lungs, the color is turned black, right? The patients, they might have carbon pigment accumulated in the tissues. As you know, one person might have different things accumulate in the lungs. Even every one of us today, we breathe in, smoke, pollution, and so on. Our lungs, my lung might also have, black pigments here as well. So, because this is from real patients, you might observe different things in the lungs. They might have different lesions in the lung. But this one, what we would like to focus is the fungal infections. As you can see, they label just fungal infections because it cannot be specified through just the gross specimen. So you have to look under the microscope. Again, first you start to look at the slide at the low magnification first, and then you observe throughout the tissues. You see where normal area looks like, the lungs, you expect to see alveoli, right? Like a small sac filled with air, right? So then you can notice which is the abnormal area. So you can tell the areas around here that the color is more pinkish than normal, and it's lose the normal character of the lung, right? Instead of having like small air sac like this, So you have like a staining of the pinkish area. So you can tell it at the lower side of the picture as well. So it's also an abnormal area. And you can tell from the low magnification that you might have hemorrhage here. Red blood cell, right? Outside of the vessel. So we call it hemorrhage. But if the red blood cell is still inside the vessel, so we call it congestion, right? So we might have hemorrhage here. You might have something abnormal here, so you have to look closely. Zoom in, so then you can see what actually happens here. But here, can you see the filamentous fungi growing? The filamental fungi growing here. Actually, in this area as well, but it's not really obvious as this one is H&E staining. So remember that when you look at fungi, especially for Aspergillus, under the microscope in the tissue, you are expecting to see hyphae of the fungi growing, so filaments structures. You might observe septations, so dividing between the cells of the fungi. So it's elongate, it's filamentous, and then you see septation, divide each cell. And then you might also observe the dichotomous branching. It's branching like a stick of the tree, and it's branched into a Y shape. So that's the character of Aspergillus. This one also aspergillosis. If you here might not look really obvious, but here also fungi, like fungi is growing. This one also as well, and this one, the whole thing, is a like bunch of fungi growing. And as I mentioned in the lectures, that fungal ball, we use the term to describe the fungi that grows in the lung cavities, right, into like a ball. We can also use fungal ball term to explain the fungal colonies growing inside the lung as well. When you look under the microscope, you can see these circular things inside the tissue. These are fungal ball as well in the scale, like under the microscope. And to observe the fungi more clearer, we use silver stain. Silver stain will stain the fungi in black colors. So you can observe the dichotomous branching and filamentous fungi more clearer in this way. Candidiasis. This is the Candida infections. Candida is usually yeast form. Candida, you know yeast, right? You might observe budding of the yeast in the tissues, but as mentioned, usually infections of Candida, it happens when the balance is shifted, right? Balance of the normal flora is shifted. For example, when you use antibiotics, it kills bacteria and fungi is overgrown. When the fungi is overgrown, the population is like the density is higher. It will turn on the quorum sensing of the pathogen. When it senses that the population of its own kind is increased, it will switch from yeast form to hyphae form. So instead of growing as a yeast, budding into like small cells, it can grow in a hyphae form. So now you can see in the slide when you zoom in, you can see both filamentous form and also the yeast form of the fungi, of the Candida. But in the case of Candida, it's usually formed like a pseudo hyphae. So you see filamentous structures, but no septation because it is pseudo hyphae. Okay. Next one is the parasitic infections. We have colon amoebic colitis. This is the colon. It might be really hard to observe, but you look for ulcer. Again, maybe really hard to observe. It should be in the lumen side of the colon. And when you see the real specimen, you might see thickening of the colon wall due to the inflammation, due to the edema. Okay. When we look at it under the microscope, remember the flash-shaped lesions, the ulcer that the neck the upper part is small, but it actually invade deeper and cause damage of the tissue, broader area in the lower part. So it has, this is the lumen surface, right? This part is the facing the lumen of the colon, and then it has small entry here, narrow neck here, and then it's wider at the end, like a flash shape. So when you zoom in and look at this area where the tissue destruction occurs, then you look for trophozoite. So trophozoite, the size is bigger, this one, when compared to our cells, when compared to our red blood cells, it's bigger in size, and it has nucleus. So nucleus, it has like dense of the genetic materials around the nucleus. So at the edge of nucleus, you will see high pigmentation at the edge, and maybe the dot spot in the middle. If you remember, you can look at it in the lecture slide. So this one is the trophozoite in the high magnification, but the above figure here is the low magnification, so you can see multiple of trophozoite there. So this one also trophozoite, this one as well, this one as well. Bunch of them there. In this area as well, this is also trophozoites. Okay, so next is the viral infections, and we also have parasitic infections here as well.
Thank you so much. Okay, thank you so much. In the station 3, we let you know about the specimen of the virus, viral infection and parasitic infection. Viral infection that let you know about the rabies virus. Okay, this show if of the hippocampus area. This is and then when you zoom into this specimen area, you can see the in EPG is iso sorry iso sorry is in another name naked body, naked body, and then it show in another name esophageal, not esophageal, naked body, but this is to show this is inclusion of the protein that conjugated with the nucleic acids, such as the ribosome that released from the or produced from the rabies virus. You can see like a pink the color of the neuron, especially in the hippocampus. But another area is in the Purkinje cell or Purkinje fiber in the cerebellum. This is involved with the destroy the neuron. Okay. And another specimen involved with the cholangiocarcinoma that's caused from the infect with the parasites, Ophiuchus viverrini. This will show the gauze specimen of this tumor mass. Okay, you can see in the nearly of the tumor mass, you can see the dilate of the bile duct. You can see this dilate of the bile duct because this bile duct has a composition of the parasites accumulate inside the bile duct and then leading to the obstacle of this duct, and the bile fluid cannot flow through this duct and then accumulate more and more and then increase the pressure, fluid pressure inside this duct. And also another way is induce the epithelial cell or that lining on the bile duct will be inflamed, and then it destroy that cell and then into the repair process and more, and then if the chronic infection. Chronic infection. This is the process from the inflammation, cell death, and repair cell. This is the cycle more and more and more. This is the repair process. It induces some gene to mutate and then leading to the cancer or carcinogenesis that look like this mass. That's cancer mass inside the liver. Okay. Okay, this is the histopathology of the liver tissue. You can see the low magnification. This is the area of the infection with the Opisthochis viverrini. And then when you zoom into this tissue, you can see this parasite in here, and the surrounded here you can see the lymphocyte. Lymphocyte, because this is the chronic inflammation or chronic condition. Therefore, it should be the lymphocyte to accumulate in this region. This is the keyword for this lab. You can see the parasites, you can see the lymphocyte, and then you can see some fibrosis inside this tissue. Okay, maybe this is for the station 3. Okay, thank you very much.
So, before, I think you have already get the worksheet very quite a lot, but just would like you to just take a photo by yourself and also label what you see from under microscope or even gloss, because some gloss specimen also different from whatever I show you here. So please take a look, and also we also have the link to submit. Have you already get it? Okay, so after the class, I don't want you to have the assignment. So try to finish in the class and also convert, export into PDF or even picture. You also can submit here. Just scan here and also submit. And please submit with this pattern, student ID and also your name and surname. Okay, so I just set deadline for the next 2 days, but I think you can finish within today. And please submit it. And okay, so are there any questions? You can take a break for a while and then you can move to the lab room. And if you have any questions, even in the lecture or the lab just now, you also can ask us at the station.ใช่ไหมตรวจปากใช่ไหมที่เห็นอุปกรณ์เครื่องไม้เครื่องมือ